Welcome everybody. Now the talk by Erich Wanker on a binary interactome mapping approach.
Okay, can you hear me? So first of all, thank you to the organizers for inviting me and giving me the opportunity to speak at this really exciting meeting. My lab works on protein-protein interaction, on protein-protein interaction technologies now for quite some time. And in this talk, I want to focus on various approaches, various efforts we have to generate protein-protein interaction data. I want to speak about limitations we currently have in terms of the data sets, and then also give you a few examples about interesting proteins or interesting protein-protein interactions that we work on in the lab. I think it is clear, and it doesn't need much of explanation that proteins do not function in isolation. They rather have to form complexes, protein complexes, or various types of complexes in order to generate function. And I think it's clear from what you already have heard, if we want to understand how a genotype gets converted into a phenotype in a cell or in a whole organism, we have to understand the underlying molecular networks much more comprehensively as we do this today. And this is important, and this brings us to the quantification or to the measurement of protein-protein interactions. And if you think about this, this is not a simple problem, or it's not a simple issue, because you cannot have a machine now where you put in all your cells or your cell extracts, and at the end you get protein interactions out like a sequencing machine. The issue is that protein-protein interactions are very diverse. They have very different affinities. You have very stable protein complexes where a complex resists even hours, and then you have very transient interactions. You have a lot of regulation in terms of protein-protein interactions. So it is definitely not something where you have one easy method and you will get them all. And specifically in the last years, from the Vidal group and from others, it is clear that protein-protein interaction networks are dominated by weak, very transient interactions that are definitely very hard to catch or to identify. And this is one issue we have to face as a field, I think. But there is also another important point that we have learned from previous studies, is that genetic mutations or missense mutations that make subtle changes. So basically one amino acid change in a protein is sufficient to change the interaction map, to change the interaction situation. So you will have a gain of interactions through certain missense mutations, but you will have also a loss of interactions. And this brings us to the question of annotations. We are still used to map these proteins or protein interaction onto the gene level. I really think we as a field need to go and map and annotate this interaction on the amino acid sequence level to be on the right, to go into the right direction. So in a way, which is currently not the case. And I think you all know we have protein variants, we have fragments, we have a lot of points here. So I think this is something we have to do as a field. We have to come to a new way of annotating protein-protein interactions. Just to give you what we are interested in the lab, we focus on mainly we focus on genetic protein-protein interaction detection assays. And this includes the yeast-2 hybrid, this includes various type of genetics tools. And the essential issue here is that we that we use tagged protein, we express them in different cells, and we are measuring these protein interactions indirectly, either by measuring the energy transfer, like bioluminescence energy transfer, or by the yeast-2 hybrid, where we have a reporter gene that then gets quantified. And this is a bit different from the biochemical methods, where proteins are enriched via antibodies or affinity tags, and where you at the end get some sort of protein composition data. You don't know exactly who is interacting with whom. You can never be sure whether there are direct contacts. And just to give you an idea what we do, we have in the lab, we have generated or developed an automated yeast-2 hybrid system where we have basically automated all various steps from the data generation to the analysis. We use picking and spotting robots, and with this technology we can now test about 70,000 protein interaction pairs per week, and we have a library that contains about 17,000 human cDNAs that is arrayed and can be systematically screened for protein-protein interactions. And just for the people here, what is the principle? How is this possible to do? We use a method that is already was quite old, which we call interaction mating approach. So in a way, we have two different yeast strains, an alpha and an A strain, and we express the bait and the prey protein in these individual strains, and this allows us to make a matrix which then by just mating this, you get diploid yeasts, and then you score for protein-protein interactions. And when you do this in an automated way, just that you get an idea how this looks like. We use quite large agar plates where we spot on the yeast cells, and here each dot is a yeast strain which contains a pair of potential interacting proteins. And you can see around 1,000 of these yeast colonies, and this is where you just control that more or less all of them grow on the agar, and then you basically spot this onto a selective plate, and only protein pairs that contain interactions can grow. And when you do this, and this is what we do in a very systematic and automated way, you have imaging. We have imaging software tools where you just automatically score or identify interacting proteins. We have a database, and then at the end we have software tools, as many of you already have seen, which then converts this binary interaction data into protein-protein interaction networks. And this works more or less in an automated or a semi-automated. We have to always check by hand or so to at the end have this high quality data. And just an example from a study we have done quite recently together with Rodet. We generated an interaction map or a protein-protein interaction data set that we call NeuroNet, where we had the idea of finding links between proteins that are involved in different neurodegenerative diseases. So starting with Alzheimer's, Parkinson's, Huntington's, this is various neurodegenerative diseases. And what we found was that indeed on the protein level, if you generate all this data, you see links that occur between, for example, Parkinson's disease and Huntington's disease, or even ALS, in terms of the involvement of certain proteins in various diseases. And when you have this data, you can run network propagation algorithms like the one that was developed by Rodet. So in a way, you start with a certain amount of proteins that you use as seeds, and then you propagate the network, and at the end you get highly connected protein clusters that are reflecting certain diseases or potential disease states. And just to give you an example, this is published. This is a Parkinson's disease module, as we call this, very highly connected proteins. Several proteins are known or have been found previously, but there are also proteins like this ICOSEC1, which hasn't been linked before. And so we looked at this specifically in a model, C. elegans model, and could see when we knock out these proteins, it dramatically increases the aggregation of the synuclein protein that is a key player in Parkinson's disease. Now we know that this is not just a protein that is relevant for Parkinson's disease, also relevant for Alzheimer's disease and Huntington's disease. Or another example from how you can use this network information, which is a network model for Alzheimer's disease that was defined based on the network propagation algorithm. And then we use this and ask the question, are these proteins abnormally folded, abnormally aggregate? So do these proteins lead to aggregated molecules? And we looked at post-mortem brains of patients and controls, and indeed found that a large fraction of the proteins that were defined here are abnormally aggregated in the brain of Alzheimer's disease patients. And so we came up with what we call a model or an idea of Alzheimer's disease, that Alzheimer's disease is a multi-aggregation disease. So at the stage where you have symptoms, so when there are many more aggregated misfolded protein in the brain than previously expected. There are certain candidates that we know for many years. This is tau, that this is A beta, but as we think is there are many more of these proteins. This is very relevant, we think, because if all the clinical trials to treat Alzheimer's disease have failed, and they always work with more or less with symptomatic patients. So if you perturb or if you take out a protein at this relatively late stage of disease, you might have no chance at all, because the other aggregates, the other proteins are already there. And this is what we currently propose. So you have to start much earlier, and we now know that you have maybe to start 15 to 20 years before you have symptoms with interfering of this aggregation misfolding process. So that's just an example for neurodegeneration. But now to this data we are generating with the yeast-2 hybrid. There are in my view, two very important limitations, which the field or many people in the field often don't like to speak about, which is on one hand, these data sets, the yeast-2 hybrid assay, as we do it in the lab, and you see it here, if you do it, you have heard from Pascal, if you do it in a very precise, so if you do it benchmarked way, is not very sensitive. So if you have 100 proteins, which are protein interactions, which are known, where you have literature, and you run them through the assay at the fast discovery rate of 1% or even lower, you will get a sensitivity of around 20–25%. And so that means a lot of information, a lot of protein-protein interactions with the yeast-2 hybrid method are missed. And I think even if you do this 20 times, or if you repeat these experiments, this is, in my view, an important issue or a limitation of this method. And another limitation we have when we generate these networks is that you have yes or no answers. So you see yeast growth, but you have no information about the affinity. You have no information about the strengths of protein-protein interactions. And this is why we have started already 5 years ago with the development of a new method, which we call Lucy. And this is a quantitative method, which a 2 hybrid method where we use bioluminescence based readout. And in a way, there are two things that I like about this is there is one, you get 2 readouts for each protein-protein interaction that you are analyzing. So first you measure in the cell, so you just use bioluminescence energy transfer from a nanoluciferase onto your fluorophore like M-citrine, and this is the first readout. And then you lyse the cell, and you get for the same interaction you do an immunoprecipitation or let we call look where you get another measurement of the nanoluciferase bound to the protein complex. And this is something we can do now in 384 well plates. And when you do the benchmarking with this assay, you can see this here. We have done this together also with our colleagues from the Vidal lab. We used a positive reference set and we used a negative reference set or random reference set. And when we do this analysis and say we want to have no interaction in our negative set, we can still detect 71% of the protein-protein interactions, right? So, and if you look at this, this is the double readout method now. This is the classical method, yeast to hybrid, MAP-IT. Many of these techniques are out there. It's basically all in the range of I would say 30 to 40, 50%, and we are now up here. And what I think is interesting as well, when we now go for a reference set of interactions that are structurally supported. So in a way, just taking interactions where there is a crystal structure or available for this protein complex. Then we combine this this Lucid technology with other technologies that are out there. We can even go up to almost 100%. So we tested this set, and except for one interactions, all interactions were scored. So I think we can dramatically increase the sensitivity of these methods, and that, in my view, might have huge implications if you then later on do network propagation or really look at biology. And we have, just to give you an idea, we have now automated also this technique, and you can see here this is a mammalian cells. We are not by far not at the level where we have been with the yeast to hybrid, but at the moment we can do about 1,000 protein pairs, protein interactions per week with this technology. And I think if we would go into this a bit more, and we could at least get 5 to 10-fold higher numbers of protein-protein interactions to be screened. Just because this was always an issue and Pascal mentioned it, that with yeast to hybrid data or methods, you don't see interactions that are normally found in protein complexes. And so we produced what we call a reference set for 3 protein complexes. So you have here each complex 5 to 6 proteins that these complexes are known they have been found with proteomics. There is structural information available, and so we scored these complexes with our binary method. And what you can see here is that at very stringent conditions. So there is we didn't allow any intercomplex interactions to occur. So and at the end, what you can see here, maybe it's a little bit too small, we can detect again for one complex even 90%, for the others around 70% of these protein-protein interactions in these protein complexes. So in a way, I believe that by combining binary interaction tools with mass spec-based methods, we might generate networks that are of much higher quality and biological relevance than we do this today. And just an example of a protein we work in the lab now, just also as a surprise, which is the Huntingtin protein. This is an alpha helical protein. It leads to genetic disease with an expanded polyglutamine sequence. I don't want to go into this in detail, but many protein interactions have been found for this protein. But when we map this with our new techniques, we have a set of protein-protein interactions that definitely bind to the full-length protein. We see some involvement of the protein in endocytosis and autophagy, and we get quantitative data for certain proteins. So we see clearly that at least one protein is binding with very high affinity to the Huntingtin protein. This is the HUB40 protein here. You can see this here by looking at the binding strengths. And we did mutations to disrupt this protein complex. You can see this here. We can see that if we introduce certain amino acid changes in the HUB40 protein, the interaction can be inhibited. And what was very interesting to see was that if we do not CRISPR knockouts in hex cells, we knock out the huntingtin protein, the HUB40 protein is completely gone. So huntingtin holds the HUB40 protein like some sort of a holding chaperone. And if huntingtin gets lost, the HUB40 protein gets lost, and we can bring back the protein by co-expressing the HUB40 protein again into this model. And so we now have evidence that this is a functional complex, and I'm not going to go into this, and other proteins are binding to this complex, and HUB40 is dependent on for the association of other proteins to this huntingtin molecule. And just to give you an idea what we can do with this bread-based technology, we can introduce the luciferase and the fluorophores, different regions in the huntingtin protein, and we can use these tags as a molecular ruler to measure distances. So if you add the huntingtin without the HUB40 protein, you see a molecular change in this protein, and you can monitor the potential distances, and we have structural information that fit very well with our data we have. So you can get very deep in terms of the understanding of specific protein complexes if you're interested. And another protein we work, and this is a molecular machine in the lab, it's a AAA ATPase, it's P97. It's involved in various diseases, in ALS, but also in cancer. And what was very surprising, normally this protein is a very symmetric molecule. You see this here. It's a very stable hexamer. And what we were expecting, if you just look at the nodes, that proteins bind to this hexamer. But when we looked into a bit more in detail, we found a protein that's called ASPL that not just binds to the hexamer, this is what we thought will happen, but when it binds, it was able to disassemble the complex and convert this into heterooligomers. So you need at some point, you see surprises in a way in terms of protein, and you can see this works basically because the ASPL protein forms some sort of a lariat structure which wraps around of a specific domain, the end domain in P97, and disrupts this complex. And we have now, and this is work in progress, we have now shown that ASPL is important for the binding of another protein to P97, which is U2AF2. This is a splicing, very important splicing factor, and you can see only if the ASPL protein is present, the splicing factor can bind to P97, and when we knock out this ASPL protein, we have a massive effect on the splicing in our cell model. And we can also show this endogenously, that the protein interaction gets disrupted. And just because you have heard a lot of COVID, we also did a small COVID project where we just were interested to find high confidence protein-protein interaction, viral protein. So we were not interested in the host viral interaction you've seen, but we were interested to see high confidence interaction in the viral proteins, and we used a machine learning where a support vector type of approach to classify this high confidence interaction, so interaction with a high probability. We tested all these 26 viral proteins against each other. We found 30 proteins that were found reproducibly. We then looked at one of these proteins, NSP16 and NSP10 in detail. We mapped the binding region. We could show where this protein interact. Then we did docking with in silico docking. We screened basically, I think it was 200 million potential compounds, and found at the end one molecule that binds now with reasonable affinity, and we are currently characterizing this compound and do structure function relationship analysis. And finally, just another method we developed recently in the lab, which we call selfie. And here the issue is that a lot of this a lot of the networks you have seen from the colleagues are all generated with overexpression of proteins in a model system. And that can lead to abnormal, in my view, abnormal protein-protein interaction. So I would say that a lot of the data out there that we have and we use as the basis for modeling might have interactions or include interactions, at least to some extent, that might not occur in the endogenous conditions. And so what we did here, we used a cell line which is called EHAP. This is a haploid cell line. Brenda knows this. So in a way, we use CRISPR to knock in with, we use CRISPR to knock in, our target reporters, and then we fuse the cells, we do electrofusion, we get diploid cells, and we can score endogenous protein-protein interactions. For example, again, the ASPL you have heard before with B97, you only see a signal when these two proteins interact, or we used another protein. This is TDP43. This is involved in ILS. So again, you just see a signal, and you can use these cell lines then also to screen for protein-protein interactions and do pathway analysis. So just to summarize, what we believe is that you need complementary protein-protein interaction assays to really get data that are comprehensive. And we strongly believe that by combination of affinity purification, mass spectrometry, various types of data sets, we will be able to create the basis to really understand biological functions of multi-protein complexes. And the future, I think, is really looking at endogenous protein complexes. This is something we have not there yet, but I think with CRISPR it is possible. I really believe also that the understanding or maybe we need to focus on certain processes, on certain modules to get high quality data, and then go and expand these activities. Just this is most important slide. Work has been done by Chris Secker, 3 postdocs, Chris Secker, Philipp Trepte, and also Christian Henig, who did a lot on the development of the assays, and many other people were involved. Thank you very much for your attention. Sure.
So I was just wondering about your comment about forming the agglomeration of proteins in the ultramirrors. So is it possible that basically once you have a seed, then any protein that has kind of better fold of kind of flavor would come and extend this participate? And is that testable with this type of yeast to feed with?
It's maybe not so easy testable with this two hybrid methods, but in principle you can have this propagation, right? You can have this. I think what is well accepted is homologous seeding, which is so you have a protein A and you will take in the other protein which has the same sequence. But there's also heterologous seeding, which is not so well described, and then people are still arguing. But I think especially in Alzheimer's, there's now more and more evidence that A-beta can somehow lead to the aggregation of tau, and it's also the other way around. We are studying this also in our fly models. So that's one thing. And then you have additional proteins that will maybe be recruited into these structures that can come in via another mechanism. Because just as an example, what we always see when we make these type of misfolded proteins, then the chaperone system will react, and the chaperones will go to the aggregates, and they will try to dissociate this. But when they are stable, so they are at some point also part of the complex. So you will have a loss of function of HSP90 is super important in many processes. regulates kinases, and that will also contribute to the phenotype. So it's complicated, but in principle, I think this is what we have to look at, and I think you can dissect it. We do this. We need other methods, but you can dissect it.
Just to follow on it. So it's possible, like, to have a yeast that has already a seed of this, you know, and then you've killed all this short protein and other chaperones.
Yeah.
And see if it's.
I have to say we do this with the flies. Actually, this is basically these dots you see here. This is huntingtin aggregates, right? They are light up. This is basically a Fred signal, and you can see the eyes here. You see the brain, and at a certain point it's full with these aggregates, and it recruits a lot of other proteins into these aggregates, and that leads then to a rapid death of the flies. So this is a way to study this, and we now know that even small amount of aggregates are sufficient. So we can turn on the protein expression for 3 days. We turn it off, and still the amount of aggregate will be sufficient to kill the fly. Sorry, yeah, there was one. Sorry, yeah. Yeah, so you want to?
Okay, I actually have a follow-up question to this. If you take the flags from Alzheimer's patients,
yeah,
is there any experimental technique that you can figure out, like what proteins are in there?
Yeah, I mean, we did a lot on this in our mouse models. So you can look at this by proteomics. You can cut out the plaques. Yeah. And there is a nice paper where people did expression profiles, so you can look at the transcripts that are there. You can look at the proteins, so there are various studies. The reality is that you have hundreds of proteins there.
Actually in there.
Or associated or with this aggregate. And so this is possible. This is something we do. The question is, what are the relevant ones, and how do they get there? But that's easy. I mean, in the meantime, yeah, with high resolution proteomics.
Okay, it's probably fascinating to see chaperones being trapped in there.
Chaperones
cannot do their usual work.
Chaperones are there. The whole ubiquitin system is there, right? So they cannot do it. So it's a massive. I always see it as a massive collapse of these systems, and at a certain point, it's too late to do something. This is something we have to realize, and that's why I'm always a bit worried when people do more clinical trials. We have to start very early. We have to measure the disease 20 years before you see symptoms. Then you have a chance.
Yeah. What was the assay on the readout of the selfie assay?
It's also bread, bioluminescence energy transfer. It's the same principle. I'm sorry, I didn't explain this. So you have the nanoluciferase, and then we use anion green because this is a bit even more sensitive than M-citrine, and that is all you need to see a signal. I have to say this is still a limitation again from this method. You definitely see you need a certain expression level of your proteins that endogenously expressed, right? There's definitely something where you might, if you have a protein that is endogenously expressed at a very low level, you can, right? You use the transcript levels and you can look at this. Then you might not see an interaction because it's not sensitive enough. But we tested it, so it's more work to do. What we did is the first set of proteins.
Question about the limitations of the technology. As far as I could understand, you always measure each interaction independently. Can you also measure what happens simultaneously, or is it not feasible with these approaches?
It's not. It's not. I mean, I have to think a bit more, I think, but it's not easy because at the moment I would say we cannot.
Because this would really give us a lot of information that is missing from the network.
Yeah, I mean, to some extent, you could. I mean, with thinking about this, right? You could with CRISPR integrate certain tags. I mean, I think you could do this simultaneously for a certain number of proteins that you define in your network, and then you see how they change either with stimulation or in a way. So in a way, creating some sort of a reporter system which reads the physiology of the cell. But that is doable, I think, but not on a real big scale because you're limited to the molecules you're fusing. But in principle, you could think about this.
So now I have the hat of my structural biology background. So we have seen that, I mean, most of the methods or interaction discovery methods that we're exploring are based on fusing proteins. Right? Yeah. And what we have seen is that whenever either the N or the C terminal of one of the proteins is within 20 angstroms of the interface, it doesn't matter what linker you use, or it doesn't matter what expression vector you use, you're never going to detect this interaction. So it could be that we are already pretty close at the completion of the type of interactions that these fusion-based technologies can detect. And then the other comments that I didn't get when you explored using Lucy, whether you reconstitute the full complexes. I don't know whether you meant the direct interactions between the complexes or all the potential interactions between the complexes, because then again, I mean, maybe we're hitting everything here, because not all the proteins within a complex are in direct contact with it.
Yeah. No, I meant the direct interactions within the complex. And you can nicely see, I didn't go into this, that certain, I mean, this is a regular 5 proteins and a protein on the outside, and you're not seeing this interaction, let's say, of protein A with protein D, which is on the other side of the complex. And it's all about the distance measurements we do. And one important point is that we now have to do the tagging at N and C in all orientations, right? So that's also what we haven't done before, and that also increases the sensitivity enormously.
Yes, but still, I mean, there's something like 7% of all the interactions that I mean, it's both. So it's the N and the C of both proteins that are within 20 angstroms of the interface. So these 7%, I mean, you will always miss.
Yeah, that's true. But yeah, that's why we need to talk about these things, right?
This is something we've done with Mark. I mean, I don't remember whether it's published or not, but certainly we have the data.
Okay, thank you very much.